Hello, Dr. Anusman. Hi.
Hello, yeah. Good evening.
Good evening.
So we are live.
Good evening and welcome to the second Noida Ophthalmic Society monthly meeting for this year. We were not there for the last 1.5 months. And today we are having a very interesting meeting on biosimilars. So all of us know that biosimilars, these are biological products which are produced in the cell, yeast or bacteria. And these products are similar to the innovator molecules, but yet they are different. And many of us sometimes hesitate to use biosimilars. Sometimes we think that adequate testing has not been done, adequate clinical, we have not done the clinical trials as much as it should be done. But it is very interesting to know that biosimilars are not required to undergo so much testing because the innovator molecule has undergone those trials and biosimilars are judged on different principles. What is the difference is that DCGI, that is the authority within our country, the FDA, the European Medical Organization, which certifies these products, have certain, sometimes somewhat different standards. So Dr. Prasad, he is the Senior Vice President, Medical Affairs and Pharmacovigilance at the Reliance Life Sciences Private Limited. So we have two faculty. Welcome, Dr. Prasad, and welcome, Dr. Gopal. I'll be introducing Gopal. He's a good friend of mine. I'll be introducing after Dr. Prasad speaks. So let me introduce Dr. Prasad. He is Dr. Prasad Dattatreya Apsanganikar. He is a Senior Vice President, Medical Affairs and Pharmacovigilance at the Reliance Life Sciences Private Limited. He has over 25 years of experience in pharmaceutical and biologicals and has over 17 publications on biosimilars. Sir has conducted a huge amount of studies, pharmacovigilance exercises, symposia, both domestically and internationally. And he is involved in the development of organization of pharmacovigilance systems and processes for the domestic as well as international regulations. We welcome you, sir, and we would love to get the basics right for all of us who are the faculty here and all those people who have attendees who have logged in for this webinar. We would like to know from you, what is the difference between biosimilars and biomimics? We would also like to know, all of us get confused about what regulations are there, what pathways have to be accepted before a product becomes accepted from the regulatory organization, and basically how these biosimilars are produced. So I would request Dr. Professor Goel, who is a president, to say a few words before we give the mic back to Dr. Prasad. Professor Goel.
Thank you, Ajay. So I will welcome you all to this Noida Ophthalmological Society monthly meeting on science and rationale behind biosimilars. So in this meeting, we have two invited speakers, Dr. Prasad and Dr. Gopal Pillai. So they will be speaking on developing biosimilars, and Gopal Pillai will be speaking on his experiences on biosimilars. So this meeting will be chaired by me, Dr. J.L. Goel, along with Dr. A.K. Gupta, who is Vice President of Noida Ophthalmological Society. And the meeting will be moderated by Dr. Ajay Arora and Dr. S.P. Chaudhary. So we now start with the first invited speaker, Dr. Prasad. Dr. Prasad, please.
Thank you, sir. Thank you very much.
Thank you.
I'm just sharing my screen for everyone.
Yes, sir. You can go to the presentation mode. Yes, thank you. Perfect.
Can we start, Dr. Vora? Dr.
Yeah,
Arora?
Yes,
please.
yes, yes, please.
Sorry.
Please.
Okay. So at the outset, let me thank Dr. Arora and the NOS for giving us this opportunity to be with you this evening. Through the incoming of biologics, we know that biologics are the novel medicines which are produced in living organisms or living systems or living cells. The first manufacturer who produces the biologic is, we call it as an innovator, as everybody knows, and that becomes, his brand becomes the innovator brand. Once they are off patent, then the other manufacturers who will develop the biosimilar to the innovator using the guidelines, their brands will be called as biosimilars, which is a common terminology globally. Some countries use other words for biosimilars. And one rule is there for the biosimilars. They have to be highly similar to the reference product or the innovator product. There are no clinical differences between the biosimilar and the innovator product in terms of safety, purity, and potency of the product. Today, we have seen good era of biological medicines, and even after 2020, there are more than 50 biologics which are going to be off patent, and we would see them in the market and their biosimilars in the market also. Now, depending on the size and the characteristics of the proteins of interest, the living systems or cells like bacterial cell, yeast cell, or mammalian cell lines are used for the production of biologics, as well as the biosimilars. And if you look at the size, when we talk of a monoclonal antibody amongst the biologics, it is a significantly bigger molecule as compared to the other biologics. India is already there, doctors. So India is already there in terms of the guidelines of biosimilars. India has published the guidelines of biosimilars in 2012, which have been almost 99% or more adopted from the US and European guidelines of biosimilarity, and the same have been revised in 2016 also. And with that, our apex body, which is called as the Central Drugs Control Standard Organization, where our drug controller of India is, they have developed good amount of committees and subcommittees like Department of Biotechnology, Institutional Biosafety Committee, Review Committee on Genetic Manipulation, Genetic Engineering Appraisal Committee, and others, who will guide, review, and play a big role in terms of approving a biosimilar produced in a developing country like India. If you look at the developing process of the biosimilars, they start from the gene of the target protein. Once the innovator brand becomes off patent, the gene information is open, and the cloning of the protein gene and transforming that gene into the cell lines or the living system is important. Then the living cell system is allowed to cultivate. The living system produces this protein of interest along with other proteins. Then the protein purification becomes the critical step, and then the protein is characterized in comparison with the innovator. Now, in this process, which will be adopted by the manufacturers of biosimilars, the vector used by the innovator, the cell line used by the innovator, the media and the methodology used by the innovator, operation conditions or the purification methodologies used by the innovator, all have to be identical with a biosimilar manufacturer, so that this minimizes the variations from the innovator and the biosimilarity will be established as much as possible, more than 99%. It is said that the clone to the complete product development should be done in the same facility. From the retrospective development of the gene, its incorporation into the host cell, cell line or cell system, production commercial production in the bioreactor by the recombinant process, and the purification and commercial development of the finished product, everything should be done in-house without any third-party step involved. The process is divided into two sections: upstream process, which has the choosing of cell lines and the vectors, transformation of the gene into the cells or the living cells, culturing those living cells. And once we get the protein, the downstream process will have an important step of separation or recovery of that protein, and the most critical step of purification of that protein to get only the pure and the complete structure of protein from all the mixture which is there in the bioreactor. And all this goes as per the US and European guidelines, which also have been adopted by most of the developing countries now. Even the Indian guidelines are 99 or more than 99% similar to the US and European guidelines. The cell line source, therefore, becomes a critical thing. Why? Because we are growing or producing the proteins in these cells or cell lines, the living systems. As we saw earlier, either it can be a bacterial cell, or it can be a yeast cell, or it can be a mammalian cell. So Lonza, Horizon, Novagen, these are some top names of the global players who are supplying cell lines and cells as living system for the production of biologics as well as biosimilars. Now, since we have a good amount of years of era of biologics, there are a lot of development which took place afterwards. So, for example, the master cell bank characterization or MCB characterization. So, once you have the cell system or the cell lines from the cell line source, it is very, very necessary, which is followed in the developed countries, that you characterize the cell bank also before you start the production of the protein in them. So it is done by the ICH-Q5D guidelines. For identity, purity, and viral contamination is checked with a host number of testings on the master cell bank. In terms of the further development today, the cell banking approach also needs to be very meticulously seen. You have to have a robust cell source storage system, and it is said that you should have a master cell bank. After you multiply those cells which you have taken from the source, you have to develop a master cell bank which will be extensively characterized and tested. And for each batch manufacturing, you will take a working cell bank from the master cell bank, so that for each batch, the consistency of the quality is maintained. Further to this, the development like single cell cloning, which is not many years old, a very recent one. So single cell cloning. So when you use the cells or the living cells or cell systems for which you take from the source to grow the protein by culturing them, it is very important to do single cell cloning. Means by using technologies like FACS, they deposit single cell in a single tissue culture plate. So like that, many plates and single cells are there, which are all transfected with the gene of the protein of interest. And then they are allowed to cultivate. So each cell will form colonies. And then they are analyzed by extensive testing that which is the colony or which is the clone which has produced the best quality and best yield of the protein which is being produced. And suppose the best clone or the best group of cells is selected to further form the master cell bank. So when this is done, then the whole master cell bank will have the best cells selected from a single cell, which have come from a single cell. And then the consistency of quality and yield will be absolute maintained to 99 to 100%. Single-use bioreactors. Now, this is a very recent development. Cross-contamination is one of the highest risks for the patients. Now, suppose there are different manufacturers of biosimilars. They will manufacture the commercial product in a vessel called as bioreactor, where the recombinant DNA technology takes place. So no manufacturer can have, suppose he's wanting to manufacture 20 proteins, he cannot have 20 bioreactors in his facility, because those are 1,000 liters, 2,000 liters, 600 liters containers. So when you use the same bioreactors for different products and for different batches, then the cross-contamination becomes a risk. So the single-use bioreactors, today technology has gone so far that such big vessels also can be disposable today. And today, with pleasure, I can say that Reliance Life Sciences has, it is the only one having single-use bioreactors, which do not involve any cleaning of the equipment. Only the replacement of the bioreactor is done. And this eliminates the risk of cross-contamination or batch-to-batch contamination to zero. Now, these are the upstream processes. After the pure protein, after the protein is produced in the bioreactors, now the quantity, the volumes are 1,000 liters, 2,000 liters, which contains all the cell lines, the broken cells, the cell DNAs, all the ingredients added, all the growth factors added, everything the whole system contains. And it is very important to have a proper purification scheme. So to get the right the purest protein and the right structured protein, depending on different molecules or different products, different kinds of filtration steps will be done, and different chromatography purifications will be done, like affinity capture chromatography, anion exchange chromatography, cation exchange chromatography, and others. So this becomes a very critical step in the upstream process, downstream process, to get a pure drug substance. Now, once you get the pure drug substance, now what becomes important is the protein fingerprint, as the regulators might see it, or regulators do see it. So here, you have to have print as that of the innovator. So it will include a plethora of testing, which is already laid down in terms of the guidelines of the developed countries like US and Europe, and now also of the developing countries. We do also have adopted the same, as I told you earlier, more than 99% we have adopted. So this test will look at the protein structure, post-translational modifications, drug-related, what are the different variants that are present as impurities, which are the process-related impurities which are present, are there any host cells, particles over there, are there host DNA or RNA which are coming as impurities. So, and plus the potency and function test of the protein which is produced is also tested. All this fingerprinting, protein fingerprinting, can be divided into analytical, similarity, physicochemical. Physicochemical means structure-wise, mass-wise, volume-wise, purity-wise. So full amino acid sequence and terminal amino acid sequence has to be identical to that of the innovator. This looks at the primary structure. Intact mass analysis. Mass has to be identical to the innovator, which gives the primary structure and overall mass. Isoelectric focusing, for example. This gives the overall charge, exactly charge identity, which needs to be again identical to the innovator. Then comes the different types of electrophoresis, like SDS electrophoresis, which I will just show you afterwards, some of the glimpses. Different electrophoresis and different HPLC techniques like SEC or size exclusion HPLC or cation exchange HPLC. These are the most high-end testings for purity. There is nothing beyond this. So electrophoresis and HPLC techniques, not one, more than one. So that's why they are called as orthogonal testings. And when these are done, it gives the overall purity and the overall structure in comparison with the innovator. Peptide mapping and peptide mass fingerprinting is also done for the purity. And it is each molecule and each kind of structure will have addition of different testings, orthogonal testings as per the guidelines. Even the secondary and tertiary structure of a protein in our biochemistry or chemistry, we know that a protein can have a three-dimensional structure or orientation in space. So even that is checked with the testings like spectroscopy and circular spectroscopy or spectropolarimetric analysis. That was physicochemical, structure, purity, mass, charge, etc. Now comes the biological analytical similarity. Biological means suppose the biosimilar and the innovator, whether though the purity is same, whether their efficacy is same, whether they are binding to the target in the similar fashion. So target binding assays, cell assays, in vivo assays are already available commercially. So with the era of biologics, all this commercial availability of kits is also there, whereby I can test the biological activity of the biosimilar vis-a-vis the innovator. Clinical similarity, of course, which we will not go in details in this session. So the phase 3 comparative study, extensive toxicology, phase 4 study, and the safety surveillance for 2 to 5 years. This can also come from the regulators, which we have also, which every manufacturer needs to complete. So this was the upstream and the downstream processes which need to be done for the development of a biosimilar. A glimpse of the application of these processes, upstream, downstream, and the characterization. Basically, after the pure substance is produced, pure product is produced. This is only showing you the glimpse of physicochemical and biological biosimilarity, how the orthogonal testings, which are 20 or 19 or more than 20 in number for one biosimilar. So just glimpses with respect to ranibizumab, which we are producing. So the innovator ranibizumab, if you see, Lucentis, as we all know, it is a mouse VEGF antibody produced in the murine cell lines. After they produced the antibody against the VEGF, they selected only the upper end of the Y-like antibody, as we know, as you all must be knowing, only the Fab end of the antigen binding end of the antibody. And they humanized the CDR regions or the complementary determining region, that is the regions which are binding to VEGF. So they humanized those regions so that it is suitable for use in humans. Along with that, the innovator also changed or manipulated the variable region in the FAB segment so that it increased 140 times the affinity for VEGF. This has been done by the innovator. When the biosimilar manufacturers throughout the globe will start, they will start from the gene structure of the ranibizumab produced by the innovator. Now, like few other proteins, ranibizumab again becomes a peculiar protein because the active form of ranibizumab, the active molecule, though the FAB segment is there, this, it is not present or it is not active in an unfolded fashion. It is active in the folded fashion. So I want a folded protein from my system to be produced. So I should use the cell lines which will directly express the folded protein outside. If I don't use that system, then I will have to do extra steps of cell lysis and add more chemicals so that I can do an extra step of folding the protein. And if your protein, your cell lines can express directly the extra folded protein, it will avoid extended contact with the host proteins and endotoxins, and it will avoid harsh agents which can cause protein modifications and lead to ADR afterwards. So the comprehensive development of ranibizumab has been achieved at our end by special cell lines with expression of folded proteins directly. The master cell bank characterization, two-tier cell banking, and single cell cloning is well established within our processes, and we are using it for all the biologics, including ranibizumab. And it's a pleasure to say that Reliance Life Sciences is the only one which today has the disposable bioreactors, which make the batch-to-batch contamination absolutely zero. We have four-step chromatography purification for ranibizumab, and whatever tests we have seen, the physicochemical and biological, we are utilizing 20 orthogonal tests for the biosimilarity testing vis-a-vis the innovator. Just one or two glimpses of this biological testings. For example, this is sodium dodecyl sulfate electrophoresis. This electrophoresis is showing that the different bands of the different batches of ranibizumab produced by us have this are occurring at the same level of the marker and having the same width and intensity as that of the innovator's band is seen in the second row. So this is one example of one electrophoresis. Another example is that of the purity. As I told you earlier, the size exclusion HPLC or the cation exchange HPLC are the very high-end testings for purity. There is nothing beyond that. So this is the depiction of the size exclusion HPLC. The point at which the innovator molecule peak comes, that is called as a retention time. So the retention time of the biosimilar and the height of the peak of biosimilar has to be exactly overlapping that of the innovator. So these are four batches of our ranibizumab compared with the black peak, which is of that of the innovator, Lucentis. Biological assay examples. Now, purity is proved, everything is proved. What about the efficacy? What about the target binding? So VEGF is the target of ranibizumab. So I have commercially available microtiter plate wells kits which are coated with VEGF, and I can test with increasing concentration of the biosimilar and the innovator how the increasing binding to VEGF takes place. So this curve shows the same thing, and the curves are curves of the innovator and the curves of the different batches of our ranibizumab are matching. And this is one of the interesting tube formation assays, and this is the most interesting in terms of ranibizumab, which I feel, because today it is available. I get endothelial cells kit or cell-based assay, which uses endothelial cells from the umbilical cord. They are given the cue to form tubes, like as if it is new angiogenesis, new blood vessel formation. And in vitro, the tube formation is done with the addition of VEGF. And then different concentration, increasing concentrations of the ranibizumab, biosimilar as well as innovator are added. And the tube formation, that is angiogenesis like tube vessel formation, should be reduced or inhibited by ranibizumab because it is going to block VEGF. So on the right-hand side, the lowest concentration of biosimilar and standard, that's innovator, is seen. And as you increase the concentration, the tube formation is inhibited in the similar fashion or same way by the biosimilar as compared to the innovator. So a beautiful tube formation assay. Such is the availability of the commercial, plethora of assays for biologics or biosimilars. So to conclude, doctors, biosimilars are entity-based, regulatory-based, today absolutely established in developing countries like India, market-based. As I told you earlier, the US and European biosimilarity guidelines have been adopted more than 99% by our country. And we at Reliance Life Sciences, we are fully integrated from R&D to marketing. We have the largest number of marketed biosimilars. These are the 21 molecules which we have already there in the market. And in terms of number of biosimilars launched, we are leading globally. Largest manufacturing capacity in India we have. And all those protocols, the important future developments which were mentioned earlier, we have the protocols of master cell bank characterization. We have the protocol of two-tier cell banking, single cell cloning, direct expression of folded protein, as we talked. So our cell line are the cell lines which are directly expressing the folded ranibizumab outside. I don't have to do any extra step, which reduces the burden of endotoxins and which reduces the burden of a protein modification. And we are the only ones, as I told you earlier, having disposable bioreactors where the contamination becomes zero. So thank you, doctors. Thank you very much for the patient listening. And I will await for the question and answer session as we have scheduled it after our respected second speaker. Thank you very much.
Thank you, Dr. Prasad. Can you unmute? It
Yeah.
was a wonderful presentation. I will request Professor Goel, Dr. Professor Gupta, and others to ask you questions. Then I'll shoot my question, my doubts actually.
Dr. Prasad,
Yes,
apart from Reliance, what are the other companies who are making ranibizumab biosimilars?
we know in India,
India.
in Intas is the one.
Yeah.
And the innovator is also selling in India, as you know. Novartis India is selling the innovator's brand.
Okay. Okay. So are they in any way superior to the basic molecules, the original molecules, or they remain as good, or they are inferior to the original molecules?
So today the biosimilar guidelines and the manufacturing is established so very well that anywhere beyond 97-98% biosimilarity is achieved, okay, by any biosimilar manufacturer. Only thing is that he has to take care in terms of the processes, as I told you. For example, master cell banking, or for example, single cell cloning, for example, using disposable bioreactors, choosing the cell lines which will express the folded protein. So if those things are not then some kind of deviation will take place, and that will bring them to say 97-98% of biosimilarity, not 99% or almost more than 99% of biosimilarity. But no biosimilar manufacturer will be superior to the innovator. Okay, they will be biosimilar. Okay. They can be identical. They can have almost 100% biosimilarity. But if the biosimilar manufacturers will produce a better molecule than the innovator, then we are going into a future era in which you will hear about bio-betters. So if a biosimilar manufacturer is able to produce a better molecule, then the next science waiting for us is bio-betters, which is not yet established properly globally.
Dr. Prasad,
Sir, in your last...
Go ahead.
In your second last slide, where you showed the tube formation, so in that every bar of the biosimilar showed better results than the biologic. So how do you?
So that standard deviation is definitely mentioned in the guidelines. Okay, so if that deviation is within the guidelines or within the norms of the guidelines, then it is not considered as significant.
Okay.
Anupma, I think the guidelines are it has to be above the 95th percentile. So if it is above 95 percentile, it is within the guideline. And I think why Dr. Goel probably asked this question was because we have these studies in our, which is not related to biosimilars. We have inferiority studies which shows a particular molecule when you compare, supposing aflibercept with ranibizumab. Then you have those studies, but in biosimilars, the word inferiority is probably never used. I have a question, Dr. Prasad, is that, an innovator molecule becomes available for a biosimilar production 20 years after the patent expires, 20 years after the first patent, isn't it?
Means the biosimilar becomes available after 20 years. This is what you are saying, sir?
So let me ask you a question. The technology that was available 20 years back has progressed rapidly beyond that. Now, for the manufacturer, say Reliance, has better technology than what probably Novartis or Genentech had 20 years back.
Yes, I can.
Can you say, so a lot of people have been talking that actually the biosimilars could eventually become bio-betters.
Yes. Dr. Arora, you are very right. But today, the bio-better as a concept is not established globally also. So it is getting established. So now you will see in near future papers on bio-betters now. But what I can say about what your question is that the innovator has done this R&D, and they have also prepared the commercial batches in the cell lines. Okay, definitely in terms of cell lines, well characterized cell lines are now available for the biosimilar manufacturers. Okay, so the innovator that time had to take utmost care and do a good purification scheme, because his cell lines were that time cell lines. Now cell lines characterization has improved very much. Those days cell lines which will express directly the folded protein outside were not available. Today cell lines which are directly expressing folded protein outside the cell are available. So as you said rightly, biosimilar manufacturers, after those 10 years or 15 years or 20 years, definitely are at an advantage of improved technology. But where they start from is the gene of the innovator, gene of the protein produced by the innovator. So earlier R&D they are not doing. Okay, so when they start from the gene of the biosimilar of the innovator product, what maximum they will reach is the 100% of the innovator product. Okay, so this is what is happening now. And now when the systems will improve further, and suppose the biosimilar manufacturers are able to produce with improved technology a molecule similar in structure, 100% similar or 99% similar to the innovator, but better in some aspects, then that is how the bio-better as a terminology is getting established. Sorry.
Another, yeah, please go ahead. Please, Dr. S.
I wanted to ask this, like bio-betters, you mean to say that bio-betters will take same time to manufacture as innovator molecules? Because here we start with the ground zero and started with new molecules.
The process will not differ, Dr. Urvashi, because what will be the starting point for the biosimilar manufacturers will be the gene of the target protein which has been produced by the innovator. Okay, now using an improved technology, we don't know after how many years that will come. Okay, so suppose the technology improves, suppose the purification methodology improves, okay, then maybe efficacy wise, safety wise, and purity wise, a molecule which is slightly superior to the innovator may be developed. But today that terminology is not in place at all in any guidelines. Okay, so since only because Dr. Goyal asked that kind of question, I mentioned biobetters, but that is definitely a future concept which will come to us. It will take a good amount of time.
You mentioned expression of folded proteins. So is it is there anything else which ensures safe immunogenicity, or the folded proteins are good enough for ensuring the safe immunogenicity?
I will.
Immunogenicity has always been an issue with the biosimilars.
Yes. In terms of purification, see, guidelines have mentioned the chromatographic purifications and the separation of the protein once the protein is produced. Okay, that purification becomes a very critical step. And the same kind of purification technologies will be used by all the biosimilar manufacturers, right? So the process-related impurities will be taken care of. Okay, but now suppose the cell lines are producing unfolded proteins. Okay, so those cell lines producing unfolded, that means I have to increase my steps. I have to break the cells. I have to break the inclusion bodies, take out the protein, and do an extra step of folding them. Okay, so that is exposing the protein. That is exposing the protein more for extended time to the endotoxins of the host cells, to the DNAs of the host cells. Okay, and then adding harsh chemicals which will cause, because folding cannot be done just like that. So some chemicals which are called as chaotropic agents are used for folding the protein as an extra step. So these chaotropic agents can again cause modification of the proteins. So the endotoxin exposure as well as modification of the proteins, okay, will be responsible for are the most important aspects of the immunogenicity or the reactions which we see, or inflammatory reactions which we see afterwards.
Dr. Prasad, I think you very well described process how we make the biosimilars. We have been using in retina for last 4 or 5 years. In fact, I just want to discuss about 2 points. I think you will tell us about this. That one is the variability in the efficacy, and second is the risk of inflammation and immunogenicity we are talking about. How often you find, because in practical as a scenario, what we are seeing, we are treating the patient, we find sometimes the efficacy varies. Although we don't see with the innovator molecule, like if you see the Lucentis, the consistency is same. But when we are using a biosimilar, somehow we have that feeling that either the variability is there in the efficacy, or we are like in the beginning, we are scared of the immunogenicity also. So do you find it can happen with the biosimilars because we are making it a different way?
No, because again, it takes me to the developing process and the characterization vis-a-vis the innovator. If these processes which I showed, the upstream and downstream processes of purification and the characterization is maintained well by the biosimilar manufacturer, okay, there should be no difference between the efficacy, safety, and purity of the biosimilar and the innovator. Okay, and I can talk about our brand, sir, because what we have seen for our brand is we have not seen any kind of chamber inflammation reactions with ranibizumab. Means our brand of Reliance Life Sciences. It has been accepted very well in terms of the purity and efficacy. We give the credit somewhat to the cell lines which we have chosen, which are expressing directly the folded proteins, and definitely the establishment of the whole upstream and downstream process in our facility. So the whole clinical study which we did on more than 120 patients and 189 patients, sorry, and the phase 4 study which we have done for ranibizumab have not given us any case of endophthalmitis or any kind of severe inflammation with our brand. Of course, we will try to we ensure maintenance of all the protocols of characterization and purity, and definitely we would take the feedback from you and other colleagues also.
Dr. Prasad, I have 2 questions before we wrap up. In case of one is, so you said you've taken the same cell line, the purification process, etc., of the innovator molecule. Is this information freely available? That there's nothing secret or private by the innovative people, that you have to discover these yourself, or they are freely available? That is one. Second, because these are biologicals and you're using the cell lines, there can be differences both in the innovator molecule as well as the biosimilars. But we always are scared that biosimilars are not as good as innovator. So are there differences over the period of years when a particular cell line is used, that there can be some changes in the drug that is coming out of either the innovator or the biosimilar cell line?
So whenever a cell line becomes available or validated or approved, okay, it goes through a very stringent process and characterization. Okay, and that is a very big characterization protocol, because those cell lines are going to be given globally for even the biologics, even the innovators production as well as the biosimilars production. Okay, so the cell lines, once the innovator molecule goes off patent, all the information of his cell lines used, all the information of whether his cell lines are expressing the folded proteins directly or not, all the information of his protocol used in purification, everything becomes available. Okay, so that is how the biosimilar pathway is initiated by the biosimilar manufacturer. So we know that even the innovator in this case, in ranibizumab case, is having cell lines which is expressing folded protein. But those times, the cell lines were expressing the folded proteins in the periplasmic space of the cells. Okay, so they only have to break the cells. Today, the cell lines are expressing the proteins outside the cell, folded proteins directly outside the cell. I don't have to do any thing to the cell. I just have to purify it. So that kind of technology advance has occurred, sir. So, but anyway, starting point is a gene of the protein. So whatever purest we will prepare, we will be reaching near to 100% of the innovator product itself. We cannot go beyond that. That will that only time will tell if the technology further improves.
So I think the take home would be that we should be rest assured ourselves that the biosimilar molecule is as good as the innovator molecule. Yeah, Ruchi.
Absolutely, sir, absolutely. See, sir, we have seen 21 biologics launched in Reliance Life Sciences. I'm also with them since last 70 years. And in terms of the quality, if the quality protocols are maintained, the biosimilars today, I can give you an example of Apiximab. Okay, Rio Pro was the brand name of Apiximab, which was of Roche, and they were totally they had captured the market. Roche, of course, when the developing countries develop the molecules biosimilar to them, the innovators will react. Okay, and we have seen the innovators reacting in many examples, in many cases. Okay, but fortunately, not only one innovator. They will definitely come back to us, and they will try to target the biosimilar which is produced by any other company or any us or any other company. But it was not identified that any dissimilarity was seen in these biosimilars. So when you have the protocol in place and the data in place, if today Apiximab, for example, only our Apiximab, we are the only manufacturers of Apiximab. The innovator has gone out of the withdrawn from the market of because many such things keep on. Of course, their priorities will be different. Okay, but we have seen the innovators reacting, and we have seen that our data being sufficient. So we will try our best to maintain that.
Ruchi, your question, and then we go on to Gopal.
Good evening, sir. I think I just have one final question. I think this is for the session. When I saw the list of biosimilars that Reliance is making, there is a mention of Bevacizumab also.
Yes.
And still in India, I think Avastin or Bevacizumab is the most commonly intravitreally injected drug, although it is not FDA approved and it is still not approved.
Yes.
Is it possible for you to come out with a single dose Bevacizumab, because that will be cost effective and safe? Safety is the major concern for.
Yes. So most of the Bevacizumab manufacturers, as you said it rightly, they are having the data where Bevacizumab is being used in AMD and other indications more than molecules like Ranibizumab. Okay, sometimes, but today even our protocol for the clinical study of one single dose Bevacizumab for intravitreal use has been submitted to the drug controller's office. Okay, and we have already developed the single dose product. We are into doing the study in AMD now, which will we expect that our Bevacizumab in Ophthal might come up by the second half of this year, or maybe at the end of the year.
That's good news, I guess.
So thank you very much. That was very exciting. If you have any more questions, I think Dr. Prasad is going to stay back with us till the end of the session.
Yes, sir.
And we can always ask him questions. So it's my privilege to welcome Dr. Gopal Pillai, a very good friend. Gopal, you're muted, so you can unmute yourself whenever you want. So Gopal is the professor and head of the department and chief Vitreoretina service at Amrita Institute of Medical Sciences, Kochi. He is an eminent vitreoretina surgeon, not only in South India, not only in Kochi, but all over the country. And he is a great human being. He's upfront about all his discussions. If he doesn't like something, he'll tell you about it in a very nice manner, without without disturbing people's peace of mind. So Gopal graduated from Thrissur Medical College, did his post-graduation from RP Center in year 2000, followed by, he was awarded FRCS. He is a member of the National Diabetic Macular Edema Task Force, member of the National Optometry Curriculum. And Gopal has been the driving force for the Cochin Ophthalmic Society. The Cochin Ophthalmic Society has become almost like a national movement for during the COVID time, and they brought out such wonderful videos that when Gopal and Cochin Ophthalmic Society were developing their programs, we were starting out for Noida Ophthalmic Society, and we have actually been stimulated. I have personally been stimulated by what Cochin Society has done. So Gopal has over 300 national and international presentations, 4 book chapters, when multiple international and national publications. So welcome, Gopal. He has also great experience in using various biosimilars. We would like to hear his experience, and then we'll bombard him with questions. Welcome, Gopal.
Thank you very much, sir. Am I audible?
Yeah.
Thank you very much, sir, for the kind words of introduction. It's always a pleasure to be with friends, Ajay Arora sir, Saspi Chowdhury, Gupta sir. Thank you very much for inviting me to this. So actually, I would say that Ajay sir has brought in a very, 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 very important topic for discussion today, because biosimilars right now are bursting through the market. Like it is not like last year. Now this year, it has been a really amazing journey for all the biosimilar companies in India. Generally, Indian pharma companies are doing extremely well. India is now currently deemed the pharmacy of the world. So that tag is really, very, very needed also, because Indian economy is growing like that. And thanks to all these pharma companies, I mean, Airlines, see the way they have done. They have how many biosimilars in the market, and that have replaced the innovator molecules as well. Really amazing. So thank you very much, Ajay sir, for bringing this wonderful discussion. Let me go to my presentation. I will talk about my experience with one particular biosimilar today. Is my screen visible, sir? Yeah. So I had some introductory slides, which I will cut off a bit because Prasad sir had in detail spoken about that. But just to go to the initial bits, because these are important. What is the difference between a biologic and a biosimilar? Biologic is any class of medicine in which the active pharmaceutical ingredient comes from a living organism that cannot be synthesized by chemical means generally. So it is a living organism which has to produce that pharmacological ingredient. Whereas a biosimilar is a biologic that is very, very highly similar in terms of safety, purity, potency, and has no clinically meaningful differences from the reference biologic or the innovator molecule that is already approved by the regulatory agencies. Need not be the exact copy in terms of formulation, but should show similar efficacy, safety, and quality. So that is the basics of biologic versus biosimilar. Now we have some newer terminology like biobetter, which is absolutely amazing. The question Dr. Ajay asked that, the technology 15 years back is completely outdated, especially in cell biological terms. And that would definitely bring in newer molecules, and all the side effect profile of the innovator would be known in 15 years, and that can also be fixed in the biobetters. Now let us look at some of these, European Medicines Agency or the FDA or the World Health Organization's definitions. The European Medicine Agency says that the biosimilar is a biological medicine that is developed to be similar to an existing biological medicine. When approved, a biosimilar's variability and any differences between it and the reference medicine will have been shown to be not affecting any safety or effectiveness. So there can be some differences, but it doesn't have any effect. Now, the FDA says that the biosimilar is a biological product that is highly similar to a US licensed reference biological product, notwithstanding minor differences in clinically inactive compounds, for which there is no clinically meaningful differences. Again, no variability in terms of safety or effectiveness. Okay. World Health Organization defines biosimilar as a biotherapeutic product which is similar in terms of quality, safety, and efficacy. So here the two terminologies which come again and again are one, safety, and two, efficacy, notwithstanding the small differences between them. But why do we require biosimilars? Okay, so the cost of a biosimilar is probably much lesser than an innovator. It bypasses the discovery stage, as a therapeutic target is already known. The phase 2 study is not required here. So we know the dosage, we know the efficacy, we know the side effects according to the dosages from the innovator product. And this can be manufactured at half the time with approximately 1/10 of the cost. Okay, that's a huge difference in terms of cost. And at least one clinical study, a non-inferiority trial, is required to compare the pharmacokinetics of the bio-originator and the biosimilar. Okay, so one sufficiently large randomized controlled trial is needed. A non-inferiority trial is needed. US patents for ranibizumab and aflibercept will expire in 2020 and maybe 2023. So in the next couple of years also, we will be seeing aflibercept biosimilar and newer biosimilars which are coming up. So this is the innovator as well as biosimilar production market. Initially, there will be an analytical area in the innovator area, which goes into a much larger preclinical data, which goes into a phase 1, phase 2, phase 3, which is like an inverted pyramid. Whereas here in the biosimilar on the right side, what you can see is that by the time the biosimilar comes in, the analytical data is so much from the innovator. And so you can very rapidly move into preclinical, clinical phase 1 and phase 3 trials. So I'm not at all going into production because Prasad sir had gone into it. Now, this is one question which was asked before, like patency or patent of the drug. It's 20 years from the date of filing with additional 4 years of data privilege, which means that they need not share the privilege of how that is, what is the data that they need not share with the other people. As for FDA, it's 12 years of market exclusively from the date of approval. And for the European medicines, it is about 10 years of market. And Indian biologics, which are authorized in India using complete data package, can be used as the reference biologic. And if the reference biologic is also not authorized in India, but is licensed and have been in wider use for more than 4 years, then that can be used. All right. So the DCGI had this approval of biosimilars in 2012. And it provided guidelines to understand and comply with the regulatory requirements of similar biologics. And step 1 was to look at the reference product, and step 2 was how were the manufacturing process they would look at, and which is highly consistent, robust, reproducible, etc. Especially batch-to-batch variations should be taken care of. And step 3 is quality-based considerations, analytical and stability. So these are some of the biosimilars which are approved in India. You see bevacizumab, ranibizumab, adalimumab, infliximab, tissue plasminogen activator, rituximab. A lot of these biosimilars in ranibizumab, now this is an older one, but all these names have been changed. And now there are maybe at least 5 or 6 biosimilars to ranibizumab, which is available. And a lot of these biosimilars of aflibercept are in pipeline. And bevacizumab biosimilars are also there, lots of them in different countries. Now, there are lots of innovative companies who are looking at not only biosimilars, but, target, how to have, extended release formulations, etc. Intel was one of the first to come up with the biosimilar for ranibizumab, Razumab, and then Reliance, Lupin, Zydus, and now Sun and Cipla and Biocon are also working on biosimilars. So with that, let me come to the biosimilar which I will be dealing today, which will be Ranizurel. And let me just start by talking about Ranizurel's phase 3 trial data. So they did a prospective multicentral randomized double-blind 2-arm parallel group active control comparative clinical study to evaluate the efficacy and safety of Ranizurel in wet AMD. So primary objective is to compare the efficacy with that of the innovator ranibizumab in preventing visual loss. That was the endpoint was considered to be patients who lost fewer than 15 letters in week 16 as compared to baseline. And secondary objectives were, safety, tolerability, immunogenicity, etc. And they had 159 subjects, 106 patients with ranibizumab and 53 subjects with the reference product group. And efficacy assessment were performed at 1 month, 2 months, 3 months, 4 months, 5 months, and 6 months. And the best corrected visual acuity along with the logmar scale, as well as the refraction and central macular thickness. All right, using OCT. Safety was based on clinical and ophthalmic examination, vital signs, laboratory parameters. Immunogenicity was checked at week 16 and week 24, and pharmacokinetic assessment in 24 subjects with a 1:1 ratio as to ranibizumab and the innovator was planned to done to evaluate the systemic exposure of ranibizumab. All right. Now, in conclusion, ranibizumab has comparable efficacy profile to that of the reference product, which is innovator ranibizumab, and difference in proportion of patients who lost fewer than 15 letters from baseline to week 16 was statistically not different between the two treatment arms. Validated efficacy endpoints evaluated during the study prove the clinical comparability. So both the ones were clinically comparable. Adverse events were comparable. Both drugs were well tolerated. Safety data was the same. Immunogenicity, there was no immunogenicity against ranibizumab. There was no apparent immunogenicity. So based on this comparable property between two innovator as well as the ranibizumab, this drug was considered a viable alternative to ranibizumab innovator in patients with wet AMD. This is the product, and Prasad sir has elaborated the 4-step purification and the 9 orthogonal tests of biosimilarity, and clinical biosimilarity is given by the phase 3 study which I just described, and receptor binding affinity of ranibizumab is similar to that of the reference product. So finally, they have found that ranibizumab was identical to the reference product, and that is how they started using. Now, currently, more than 11,000 ranibizumab injections have been done. 350 doctors have used. Not a single case of adverse event or serious adverse event has been reported so far. And this is published in Clinical Ophthalmology. Now, coming to use of biosimilar in this talk, I will give you some clinical examples. The three data which I would like to give would be: one would be switch patients on innovator ranibizumab, which I shifted over to ranibizumab. And number 2 is treatment naive patients, and overall results as compared to that of an innovator. In my hands, it is not a huge number here, maybe somewhere around 20 patients in each arm in AMD, DME, and RVO. Now let us look at switch patients in these things. So this is a patient number 1 who had a wet AMD. You can see large pigment epithelial detachments with some fluid, and the logmar vision was about 0.4, and this is the OCT prior to the injection. And you can see that after, initially we had treated with Accentrix, and the vision had improved to 0.3, and then after the second injection, it had improved to 0.2, logmar 0.2. And then at this point of time, the patient was switched over to ranibizumab, and you can see that the 0.2 vision logmar continued over the next 2 ranibizumab injections as well. You can see that the fluid, the pigment epithelial detachments were there, but the intraretinal or subretinal fluid was not there much. All right. So this is patient number 2, again, wet age-related macular degeneration. Initial visual acuity was logmar 0.8, and this is the OCT prior to intravitreal. And you can see that it reduced to 0.6. You can see a very flatter pigment epithelial detachment, which is slowly reducing. And after second Accentrix, actually the fovea came down, even though there was a small pigment epithelial detachment, which is not very consequential to vision loss. The patient improved in vision significantly to about 0.2, and that 0.2 was continued with a ranibizumab injection. You can see slight waviness, but you can see that the pigment epithelial detachment has sort of reduced after the first ranibizumab. Now this is the stage the patient is in. Now patient number 3, here is a patient for switch. This is a retinal vein occlusion. You can see the large edema, about 700 microns. There was a large cystoid space and spongy, and there was a small taut hyaloid here. And this is the OCT prior to the anti-VEGF. You can see that after the first anti-VEGF Accentrix injection, vision had improved to 0.5. You can see that the macula had that huge cystoid spaces had had actually gone out. But after some time, when we did the repeat OCT, you can see that the edema was increasing again. And that is a point of time when we gave a second Accentrix. We don't give loading doses to retinal vein occlusion patients. Generally, we do PRN. So if the patient is coming back a little later, probably there will be a worsening of edema. And usually after the worsening of edema on the OCT, it takes about 2 weeks for the vision to drop. So when the vision drops, the patient comes back and usually the edema would be much larger. That's the usual situation in an RVO. And here is when we had the second Accentrix, where the visual acuity was almost the same, logMAR 0.5, but the edema had actually worsened a bit. And here we gave Ranisural, and you can see that the visual acuity actually improved to 0.3. And then you can see that further there is an improvement, and the vision is maintained at 0.3 after the second Ranisural, but you can see that the OCT is significantly better than the previous OCTs here. So this is one case in which the Ranisural proved to be a little better clinical efficacy than the innovator molecule. Here is a PDR with a diabetic macular edema with a logMAR visual acuity of about 0.6. OCT prior to this is cystoid macular edema. You can see that this is slightly chronicity, because this much of cystoid spaces will show that there is a little bit of a chronicity here. And even after injection, the edema actually was not very well controlled. But after the second Accentrix injection, the edema almost went back to normal, and OCT was also very good. And then we continued to inject after the third Accentrix. After some time, this OCT picture actually became very worse when the patient came in. And so we had to give 2 more Accentrix. And these are the pictures after that. And then we started injecting the patient on Ranisural. You can see that the large cysts have disappeared. And now it is more of a spongy edema here. And actually visual acuity also improved from about 0.5 to about 0.3 after the second Ranisural. So these are some switch patients. Okay, in switch patients, we showed that in AMD there was almost equal response, and in retinal vein occlusion we had one patient with significantly better response, and even in diabetic macular edema, which are quite chronic and a little bad, we could see very good response as compared to the innovator. Now, treatment naive patients, I had some DME patients. This is from another center. So the OCT is a little not so clear. So this is 0.5 logMAR with 462 microns post-treatment actually. Treatment naive patient after 3 injections of Ranisural. Actually, you can see that now the patient's vision has improved to 0.2 with almost normal looking fovea. And here is again another patient with 6/60 vision. There is a bit of cataract as well. You can see submacular detachment as well as cystoid with spongy edema here. And you can see after Ranisural and focal macular laser, the edema had reduced. And after focal macular laser, you can see that the hard exudates are also reducing. This is post 3 Ranisural injections. And then you can see that visual acuity is also significantly improved. After some time, it came back, and you can see that again with injections, it has significantly improved. Now, this is again another patient with a little bit of a taut hyaloid with a large edema, 550 microns with 0.6. Actually, post-treatment with Ranisural, significant improvement to about 259 microns. Normal, you can see it is almost normal with good visual recovery. So in proliferative diabetic retinopathy with vitreous hemorrhage prior to vitrectomy, also you can give Ranisural. This is pre-surgery. After injection, we have done a vitrectomy, and you can see the near normal looking macula in the central area. So these are some of the composite graphs. This is in wet AMD. You can see that the logMAR visual acuity from about 1, it reduced to about 0.4 or something like that. So over 3 or 4 months, significant improvement in visual acuity of about 6. There are significant improvement of visual acuity, and you can see this is the improvement in terms of central macular thickness here. So as compared to that in the Accentrix of about 25 patients here, you can see that there's this is the graph which is not as good as the Ranisural. Whereas in diabetic macular edema, with my current number of patients, maybe I should look at the data a little bit more to see if one or two patients are actually skewing the results to the side. So visual acuity is not significantly improved, but you can see that the macular thickness is reduced significantly as compared to, again, this is the innovator molecule, visual acuity as well as the macular thickness has actually reduced. This is retinal vein occlusion, a result in ranitidine. You can see about from 1.2, it is reduced to about 0.6. And this is the significant improvement in terms of central macular thickness with ranitidine. This is over a 3-month period, and as compared to Accentrix. Accentrix had more patients in the study because we have been using Accentrix for a long period of time. All right, so VRSI had conducted a survey of which Dr. Ajay Arora is a major part. He was the person, I think, who conducted this survey. About 112 members participated in 2018, and in 2020 about 100 people participated. And both surveys, the majority respondents were aware of biosimilars. Nearly 100% people were aware of the biosimilars, and there was an increase in the ranibizumab biosimilar from 41% to 56% in the 2 years between 2018 to 2020. If you are doing it now, probably that number will be close to about 70%. And 2018 to 2020, the proportion of respondents satisfied with safety had increased from 61 to 68%, and efficacy had actually improved from 65 to 81%. And I hope that in these 2 years, between 2020 and 2022, when VRSI may be doing one more survey, actually the safety and efficacy perception of people would have increased better. But we also noticed reduction in satisfaction levels with the bevacizumab biosimilar as compared to Avastin. So to conclude, the VRSI survey reveals that Indian vitreoretinal specialists are familiar, and there's a progressive trend favoring ranibizumab biosimilar. 1/3 of participants deem the current price of ranibizumab biosimilar to be appropriate to replace Avastin. That's about 1/3 of the participants. So, but we needed enhanced pharmacovigilance and larger clinical trials. So to conclude about biosimilars are the need of the hour. Basically because in India, the number of injections per person per year is not as high as in the Western population. Basic problem is about affordability. And if the efficacy and safety are as good as innovator drugs, okay, and safety was way ahead of off-label drugs and stands the legal scrutiny of DCGI, I think Indian population, since it pays out of the pocket, biosimilars are very, very important. Thank you very much for your patient hearing.
Thank you. Thanks, Gopal. That was wonderful. I would ask others, Dr. SP, Dr. Vichir, Dr. Anupma, Professor Gupta, Charu, Urvashi, if you have any questions, please.
Hi, Dr. Gopal.
Sir, I would.
Hi, nice to see you after a long time.
Yeah, long time.
Yeah, nice presentation. Just wanted to ask, like now almost all of us have shifted to biosimilars. So how much percentage of patients are using biosimilars and as compared to innovators? And as far as the Avastin and this biosimilars?
Yeah. It would vary from practice to practice and place to place, metro versus rural, or your own practice, I mean, charitable practice versus business type practice. It completely varies. So all spectrum would be there. But in my practice, generally my biosimilar usage in terms of ranibizumab would be approximately, let's say 50%. Okay, 50% would be biosimilars, 50% would be, but I'm also using Avastin. So there is no, I mean, in my place, Avastin costs about, much less than even a biosimilar. So Avastin cannot be replaced in my place. But if some places are there where Avastin cost is also very high and the biosimilar cost and Avastin cost are almost the same, then Avastin can be completely replaced. But that is not there in our state as of now.
I fully agree with that. In fact, for the last 7 years, I'm not using Avastin. Almost shifted to biosimilars, almost 80 to 90% I'm using biosimilars compared to
privilege of Delhi.
Yeah, definitely. Reason being this.
Yeah.
All your switch patients, you showed that there were better results with ranitidine as compared to Accentrix. So
not all.
Most of the switch patients which you showed in your slides.
Yeah, I showed only one, which I showed a nice response. But you see, if you look at the comparative graphs, you could see that AMD patients actually fared well with the ranitidine, whereas the DME and the RVO patients, I think we do not have regulatory approval currently for ranitidine for the DME as well as RVO. But we have been using it as an extended indication, just like we would use a Avastin off-label. And I think with the initial results that I have, maybe the innovator molecule is a little better.
I just wanted to ask you about certain refractory cases of severe DME which you showed. You didn't try any Ozurdex, or you do believe in?
I definitely try Ozurdex, and Ozurdex is a good drug in refractory DMEs. Now these are switch patients in which I switched to ranitidine, which I showed, right? So there are a lot of patients whom we switch to Ozurdex who also do well. Yeah.
Gopal, why did you switch? Because you injected
very good question, sir.
You injected two Accentrix and you switched to ranitidine. Why should you do? Why should you do?
It is. I mean,
is it because Dr. Prasad made you do it?
No. So at some okay. So the government sponsored patients moved from the innovator to the biosimilar, because the ECHs and the ESI and all moved like that. So now I had, so earlier they were giving the innovator. Now after probably some rate contract discussion with the biosimilar companies, they moved to the biosimilar. So these were like, if you want to treat, you treat with this. That is all. So those are the switches. It is not the switches that I made. The government made those switches for us to evaluate.
It was not a response related.
No. It was not a response based switch. Correct.
Dr. Gopal sir, hi. Good evening.
Good evening, Ruchir.
Am I audible clearly?
Sure. Yeah. You're in the car.
I know you're in the car.
Yeah.
You're not driving?
Sir, I just had one question. No, sir, I'm not driving. Yeah. It's not like
you're
that.
Not as bad
I
as
just
me.
had one question. No, I'm not at that level yet. So yeah, sir, I wanted to know, see, in my practice, I use more than 90% biosimilar for DME and RVOs. The only place where I'm not confident is an ROP. And that is the only place where I would always give Accentrix. So you think, is that justified, or is it okay to use biosimilar in ROP? Because per se, Rainbow trial only talks about ranibizumab of the innovator. What would be your point on that?
I think Ajay sir and other senior people also should be answering the question. This is a very serious question. Actually, in my mind, there is no difference in terms of clinical efficacy statistically significant difference between innovator and the biosimilar. So there is no reason why you wouldn't use a biosimilar in ROP. But having said that, the courts in India are not very kind to us. So you should understand that even Dr. Prasad sir will only talk to us to use ranitidine in wet AMD, because that is the one which has got regulatory approval. Now, you treated a child with ROP, and unfortunately, because of the severe nature of the ROP, you lost the eye. The patient can sue that, you used ranitidine, that is why I lost the vision. And they would look at whether this ranitidine is useful for ROP. The regulator would say, no, it is not. So I think ROP is one condition in which, last maybe a few years back, there was a big court case with 2.5 crore handout. So this is open to discussion. Ajay sir and Chaudhary sir, please respond.
Is it okay for adding something?
Please, sir.
Thank you very much, Dr. Ruchir and Dr. Goyal, of course, Gopal, of course. Now, as you pointed out rightly, that at present, we don't have the approval for the other indications apart from AMD. But only thing is that here, globally, doctor, globally in US and Europe also, in the guidelines of the biosimilarity, there is a provision for the biosimilars, even in the developed countries, to get the extrapolation for the approval of other indications once the efficacy and extended safety is established in the indication where they have done the trial. So even the Indian guidelines to which we have applied for the extrapolation of indications to the other indications, like it can be done in the Europe or US biosimilars also will have the same condition. So now they have told us for that, they have told us to do a phase 4 study. And they told us that after we get the result of the phase 4 study, we will consider giving the extrapolation to other indications. So which now we are going to submit the report of phase 4 study and expect the approval for all the indications apart from MDs.
I think I also had a similar. I just wanted to ask Dr. Prasad that in case an innovator molecules is, say, approved for 5 indications, 6 indications, and you make a biosimilar, once you have the phase 4 data, then automatically doesn't it apply that all those 5 indications become applicable to the biosimilar?
Yes, absolutely. Because what are the guidelines focusing on are whether the biosimilar is identical in terms of quality and efficacy and safety in a particular indication, and that quality and efficacy cannot be different once the biosimilarity has been proved by all those orthogonal tests and all the protocols which have been there in the US and European guidelines. So even a biosimilar in US, sir, US or Europe, suppose there is a biosimilar, even they will have this provision of once the safety is established, they will also get extrapolation of the indications for other indications.
I think there's a difference between how FDA approves and EMA approves, European Society, is that FDA has a post-market immunogenicity assessment. Initially, the company has to submit before it gets approved, but EMA and DCGI does not require a post-market immunogenicity analysis.
Yeah, because the phase 3 immunogenicity is what the EU and the DCGI talks about. And then they talk about the phase 4 study in case you are going to ask for other indications.
Okay, so I think that's been wonderful. We can now go on to the case presentations. And I suppose, Gopal, you're going to stay with us. Don't tell us that you have something else coming up. We want you there. We want to hear your comments. So we have now for this session, Dr. Gopal, Professor Gupta, Professor Goel as the chairpersons. Dr. Ruchi and myself will moderate. Of course, we have Dr. Anupma with us. So Anupma, please, these things don't matter. Please go ahead and ask questions. And we have panelists, all the presenters are the panelists. So we'll go ahead with the first case presentation by Dr. S.P. Chaudhary.
Thank you, sir. I think Dr. Gopal has already presented few cases. I'm just going to put 2 or 3 cases from my experience. I think, as I told earlier, that I have almost shifted to biosimilars for 80 to 90% of my patients. But there are sometimes when I feel that there is some like variation in the efficacy. So 2 or 3 cases, those who responded well. I hope my screen is visible.
Yeah.
Yeah, okay.
You put it in the presentation.
Yeah. Is it okay now?
Yeah.
So I just start with a few of the recent patients. One of this patient was 64-year-old gentleman who presented with blurring of vision in the one right eye. And he had all the gamut of problems, diabetes, hypertension, nephropathy, and everything. And he had actually presented with the right eye blurring of vision, which was having advanced cataract, but the left eye was pseudophakic. And it showed changes of severe NPDI with diabetic macular edema. Right eye is not visible. So this is the right eye, which showed a lamellar macular hole. There were not much significant edema, but the left eye, although he was not aware, he was using this eye very well. The vision was 6/18, pseudophakic with the cystoid and spongy macular edema. So I just went ahead with a biosimilar molecule, and this was the response within 4 weeks. So actually, these biosimilars, they respond very well. Although these most of the cases where the cystoid edema is more, they will respond well. So this patient is doing well. So I have not repeated this another injection for this, and because I am also most of the time titrating my patients and giving on PRN basis, and for EMD basis on the loading doses also. This is another patient, a 66-year-old gentleman, again uncontrolled diabetic. He came almost a year back when I advised him injection in one eye for CVR and PDR, and this time he came with a PDR in both eyes with vitreous hemorrhage also. So for this eye, like in the left eye, you can see the spongy and the cystoid edema, and the other eye, which actually I advised the injection last year, has reduced, so it subsided on its own. So I advised intravitreal regimen with 3 sittings of PRP, and this is how the response was. And vision improved to 6/9 and 6, and I repeated another injection because he was living abroad for 3-4 months. So I given another injection to this patient. Now these are 2 cases of diabetic macular edema. Beside this, their response was very good. The third patient is of a AMD patient. This is a 72-year-old female. She's a CGHS beneficiary patient. So one case of diabetes, hypertension, and because of COVID and some systemic problems, she was like hospitalized, and she was using some sterile nasal spray. And this is what she presented with 6/36 vision and 12 and with macular RP changes, lucid and shallow SRF. OCT and FFA was somewhere else. I don't have the pictures right now, which confirmed occurred seen in this patient. And this is after the 1 month after first injection. This is how the patient responded. And this is after the second injection. She responded, but the vision didn't improve much in the side. I don't know why. And I have given a third injection. I'm waiting for the like follow-up of the patient. So biosimilars, in fact, because of they were required because of the cost which we are having, the cost burden patients are having in their life. So biosimilar, they are very effective, as good as the inhibitor. Thank you.
Thanks, SP. One question to everybody. When a patient walks into your clinic and you are going to inject ranibizumab, you give options to the patient that, look, you have ranibizumab, we have Eccentrix, we have ranitidine, we have razumab, etc., etc. Do you give them the option, or you said I'm injecting ranibizumab, and do you tell them it's a biosimilar?
Yeah, the option is given only for a biosimilar and this innovator molecule, Eccentrix or biosimilar. But till now, recently we are starting having it. Maybe because of the cost differences, I have not given option for different molecules like this. Only either biosimilar or innovator molecule.
I also give option between Eccentrix and razumab, but of course the CGHS and DCHS patients, they get razumab from their this thing only, office only. So by default, we give razumab to the CGHS and DCHS patients. But the other private patients, I always give an option of Eccentrix or razumab.
What I feel that the biosimilars are not eating into the eccentric part of our patients. They are mostly for the people who are using Avastin or Biosimilars, mainly for a biolegal purposes, and mostly because we don't have compounding pharmacy like people outside. So those people who are able to afford Eccentrix will afford it later also. For us, it is a patients who are not affording the Avastin, and because of our limitations, because we don't feel safe with Avastin anymore, so we have this option. So for me, I just give 2 options right now. That is either to use Eccentrix or this biosimilar, which is razumab, and let them decide the pros and cons for both. But then the main thing is whether we believe in the biosimilar or not, and how we project it.
I think now all of us now believe in biosimilars, but I still continue injecting Avastin in some of my patients. Avastin is not out of hope for me. You have to be just very careful when you choose Avastin. So we give them all the choices and let the patient decide.
Sir,
Gopal.
Sir, I would like to say that if you are giving very many choices to the patient completely gets confused. So your objective is to treat the patient. So obviously you can say there is this, Avastin is there, which is not designed for the eye per se, but we have been using it. Then you have the ranibizumab, the innovator ranibizumab is there. A lot of Indian biosimilars to ranibizumab is there, which are almost equal to it. What do you want? Or, then you can move to aflibercept, and probably I tell them about clinical trials. Maybe in DME, I would talk about, the protocol T. In AMD, the CAT trials or the newer trials, I would definitely say that this is more efficacious than the other, but this is costing this much, this is costing this much. That would be there between molecules. But even then, people will take 1 or 2 days to go talk to people, talk to their relatives, look Google, maybe talk to some doctor some gynecologist who knows them, and he would decide for that person. So all that is there. But if you are telling this is the drug for you, then they will probably decide then. So it is about practice management also.
Yeah. In the end, once you talk everything, you'll say, okay, doctor, okay, sir, आप ही बता देना क्या inject करना. I said, I'm so sorry, I've spent so much time telling you all this. And now, so that's the way things are.
No, but that is important, I think, sir. We should have that talk. And I would definitely, in an AMD patient, I will talk about 5 years. Or I will talk about the 7-up study to them and tell that, this is what you are looking at 7 years. It is not about let us look at one injection and see. No, absolutely not. In DME, I will tell them about DRCR protocol I and say that 5 years, this is the number of injections. And we would not be as strict as them. We will have a number of injections will be a little less. Yeah.
Yeah, so in my practice, where I'm working, it's a medical college. So most of the patients come looking for cheaper alternatives. So I just give one option, that is a biosimilar by Intas Pharmaceuticals. Again, because we don't have a compounding pharmacy, so I'm not using Avastin. Until, of course, the indication is for example, a pachychoroid disease or IPCV, where I would, choose aflibercept. Otherwise, in most of my patients, say 90-95%, I'm giving the only option is a biosimilar.
Dr. SP sir, I had a question for you.
Yeah.
Have you ever felt that there was a particular case where you had to shift from a biosimilar to the innovator molecule?
Yes, there have been few instances. In fact, initially when I was talking about, somehow I felt that few of the AMD patients, they don't respond. The response is not as good as you could have gotten with the Eccentrics or aflibercept. In fact, in one of the patients, that was CGHS patient, I given for myopic CNV, I have given this Rezumab, but surprisingly, the distortion didn't go, although the hemorrhage has gone down a little bit. But when I shifted to Eylea, there was dramatic difference within 3 to 4 weeks. In fact, the case which I showed just now, the response was there, it was good, but I have given 3 injections to this patient without improvement in the vision. So sometimes the expectations are a little high, but they do respond. There's no doubt about that. But yes, these cases are where I feel that there is some variability. That's why I asked this question of variability in the efficacy. That sometimes from batch to batch might vary in biosimilars, not in the innovator molecule.
Actually, there's a small distance from what I gather. You were saying you switched from a biosimilar to aflibercept. So shifting from, just one second, so shifting from ranibizumab to aflibercept is actually a switch.
Yeah.
It is not a change from a biosimilar to the innovator molecule.
Yeah,
That's what I wanted to ask you.
So this was basically CGHS patients. So we don't have any options over there. But I was just talking about the patient was not responding to this ranibizumab, but responding with aflibercept. But in AMD, we have seen this kind of switch will make a difference. But the myopic CNV, which they respond to, even Avastin, they respond very well. But there was not much of response after that.
Yeah, because I feel with the schemes available with aflibercept, if they are not responding to a biosimilar, I would ask everyone, I would personally switch to aflibercept and not switch to an Eccentrics in such a situation. What do you think, Gopal?
Aganex?
Huh?
Aganex.
Dr.
Switch to brolucizumab.
Oh.
Sir, my question was actually a bit different. See, as I mentioned, 90% of my patients get biosimilar because I frankly believe that they work really well. But somehow, in a particular case, if a biosimilar is not working, would you give a chance to the innovator molecule or shift the molecule entirely?
No, I'll give a chance to the innovator molecule first, and then I will shift to the different.
Ruchi, the question here is the cost. The cost of eccentric is same as aflibercept now. So would I? So that and if you look at the posology of aflibercept versus posology of eccentrics, the duration of action of eccentrics is say about 4 weeks, while for aflibercept is about 60 to 70 days. It's about almost 2 months. So that way, not only I'm saving on the cost, I'm also saving on and I'm getting a different molecule which may be more effective. But yes, you are very right. You can certainly switch to eccentrics. If the aflibercept was costing the same what it costed about 2 years back, I would try eccentrics. Not now.
Dr. Chaudhary?
Yeah.
You always switch over to the innovator molecule whenever you feel not confident in the biosimilar?
Definitely.
And you always feel there is a better response?
No, I'm not saying this is what I said, that some of the cases I feel so, not all the cases. In fact, that's why I shifted to 80 to 90% of my patients are on biosimilars. But some of the patient I feel that no, this was not working as it should have worked.
And that could have been same, like it could have not worked with the innovator molecule also. Is that true?
Quite possible, but after so many years of giving so many injections, sometimes with your practice, you try to get that feeling that no, this would have responded very well.
It's a gut feeling.
I would actually agree with Dr. Ajay sir in this, because if I see, I think in my mind, it's completely debatable, and it's a personal experience. In RVO, Avastin versus ranibizumab versus aflibercept really doesn't make a difference. Significant improvement in vision, significant reduction of the edema, PRN basis, no problem, no difference. I am talking about most patients, but there will be one odd patients in whom the number of injections need to be very, very frequent, and in those patients, definitely aflibercept as posology requires that less number of injections. Obviously, that would be good. And Paganex definitely has longest duration, because you are bombarding much more number of molecules inside. About 10 times the more number of molar concentration is being inserted. Regarding DME, Avastin is there, ranibizumab is a little higher, aflibercept is a little higher as per protocol T as well. Paganex has not come up with it, but I think it is slightly better. Your duration of the number of injections can be reduced. In AMD, again, Avastin versus Accentrix, definitely Accentrix is better. And in all the non-inferiority trials that has been published so far, the biosimilar has not surpassed the innovator. It's like non-inferior, that is all. So the results are a little less, but it is not statistically significantly less. If the numbers were to be higher, like 1,000 patients, maybe statistical significance may have come in. Okay, so they are looking at some 200 patients, 250 patients to get the statistical non-inferiority to the innovator. But according to Accentrix and ranibizumab and aflibercept is much better molecule for AMD. No, I mean, it's reasonably clear about it. So if I want to switch from a biosimilar, I will definitely switch to aflibercept in terms of a AMD. But in DME, yes, if the poor visual acuity is there, definitely it is better. In RVO, really doesn't make a difference. That's what I think.
I think we'll go on to the next presentation. That's for Urvashi. So thanks, Urvashi, for joining us. I've been trying to get you onto the thing for a long time. Then let's hear Urvashi. You can share your. And after Urvashi, it will be Charu and then Ruchir And if time permits, I'll present, otherwise I'll skip.
Some issue with my screen. Can you go to the next presentation? I'll do it after that.
Okay, Charu, are you there? Yeah, Charu, can you present?
So is the screen visible?
You're not yet shared your screen. Go on to that green button below and share.
Yeah, yeah, sir, I'm doing that.
Oh. That's correct. So go on to the
Is it visible now?
Yeah, it's visible. Go on to the presentation.
A very good evening, everyone, and thank you, Dr. Ajay, for the kind invite. So I am presenting just a single case. My patient is a 65-year-old one-eyed gentleman who's a high myope, non-hypertensive, non-diabetic. The patient was operated for cataract in both eyes many years back, and he presented to me in November last year with complaints of distorted vision in his only seeing eye, which was the left eye, since past 1 week. So on his ocular examination, yeah, the best corrected visual acuity in his right eye was finger counting at 1 meters. The best corrected visual acuity in left eye was 6/9 parts N10. His intraocular pressure was normal in both the eyes, and he was aphakic. On his dilated fundus evaluation, the right eye showed a myopic fundus with an extensive chorioretinal degeneration at the macula. The left eye showed a myopic fundus with the chorioretinal degeneration, mainly peripapillary, and was sparing the fovea with a fresh foveal hemorrhage. So the OCT through the left eye showed increased retinal thickness with a central subfoveal thickness of 338 microns. There was minimal intraretinal edema and a hyperreflective subretinal membrane superior to the above the RP. So this was a case of a choroidal neovascular membrane secondary to his high myopia, and this was shown by the OCT angiography, which showed a network in the left eye. So the patient was advised intravitreal anti-VEGF injections, and I normally advise initial 2 injections in cases of myopic CNVM. So this patient initially received ranibizumab innovator in November 21. 1 month post his injection, his retinal edema reduced, and the best corrected visual acuity improved from 6/9 partial to 6/6 partial. So the patient went ahead with second ranibizumab innovator molecule in December 21. Then probably he was apparently well, so he did not come at his 1 month follow-up. However, he came 6 weeks post the second injection with a worsening metamorphopsia since a couple of days. So this was in Feb this year, where the OCT at this point showed increased retinal thickness, which had gone up to 319 microns, and the best corrected visual acuity had again declined to 6/9 partial. So the patient at this time was advised another injection. In fact, he being one-eyed, I advised him a stronger molecule, which was aflibercept. But owing to his financial constraints, the patient instead opted for a biosimilar injection. So the third injection which he underwent was a ranibizumab biosimilar. So this was given in Feb this year. And as with all injections, the first post-op day, there weren't any signs of inflammation in either the anterior posterior segment. The intraocular pressure was normal. 1 month post-injection, the best corrected visual acuity had improved to 6/6 partial. And there was a significant subjective improvement in his metamorphopsia. The intraocular pressure was normal. Patient did not report any systemic adverse event. So this was his OCT at 1 month post his first ranibizumab biosimilar. So this was the third injection, in fact, and his retinal edema had reduced and the visual acuity had improved. So at this point of time, I advised him another injection, but again, because of his financial constraints, he came after 2 months for the second biosimilar injection, which was now the fourth injection. So this was 2 months post the first biosimilar injection. So again, there were no signs of any inflammation, and 1 month post-injection, the best corrected visual acuity was maintained as 6 partials. At this time, there was no subjective metamorphopsia, and the patient did not report any systemic adverse event. So OCT at this time showed the retinal edema had nearly resolved, the foveal contour had restored. This time, the central subfoveal thickness was 290 microns, and the patient was maintaining a good visual acuity. So this patient has now been advised a close monthly follow-up. So this case is against what Dr. S.P. Chaudhary just shared. So in his case of myopic CNVM, and why I chose this case was just because one, we know that myopic CNVMs, they do better, and I normally advise 2 injections. However, this patient, he underwent 4 injections, and in fact, the first 2 injections were the innovator ranibizumab molecules. Since still he progressed, and after the 2 ranibizumab biosimilars, he is doing relatively good. So yeah, that's it. That's from my side.
Great, Charu. Good to know that biosimilar worked in this case when you were actually switching him over to aflibercept. And yes, he's doing well. So that proves the point that biosimilar probably works as well as the innovator molecule.
Yeah, so in this case, it did.
Any question? Can you unshare?
Yes.
Yeah. Any questions for Charu? Any comments? Urvashi, are you ready with your presentation?
Yes, I am.
Just let Charu unshare and then we can.
Excellent case, Charu.
Thank you, sir.
I would also want to, look at the possibility that instead of 2 injections, 4 injections did the trick.
Right.
4 injections of anti-VEGF agents reduce the VEGF levels so much more than the second 2 injections, so that the myopic CNVM died.
And myopic CNVM sometimes, they will once you've injected, they've died, but they still, however, need to be followed, and they may not need injections for a long, long time. So if you have no questions, thanks, Charu.
Thank you, sir.
Can you please share your screen? And Ruchi, please be ready after this.
Yeah, sir. I'm ready.
For time permits, I'll present.
Able to see my screen?
Yeah,
yes, ma'am.
Yeah, thank you so much, sir, for this opportunity. And there is excellent talk by Dr. Prasad and very informative also. And about biosimilars, I will share my experience about the biosimilar Razumab, which I used. Like we all know, it is similar but not identical. And the main thing is the cost. The factor which is making us switch towards this is mostly the cost. Now I'll share this experience of around 215 patients which have started using since I've used this molecule since 2018. And here I have included only those patients where I had a follow-up of around 9 months. The indications were around DME, roughly 85 patients were there, AMD 75, RVO 43, and others were clubbed into 14 patients. The all injections were given in OT under sterile precautions. Now for DME, what I found was that using this biosimilar, the vision which ranged among the patients was from 260 to 6.9 partial. The thickness was average around 400 microns, and I had to give around 4.5 injections over the period of 6 months. And the gain in the visual acuity was 6 lines in many patients, and with average fall in thickness to around average of around 30%. Some patients didn't respond to this drug after initial few injections, and I had to switch them over to Ozurdex. And this is the one case. This is a 51-year-old male with best character visual acuity was 6.12, which improved to 6.9. The CMT decreased from 390 to 283. And this is the case of severe DME. And here we can see by the biosimilar molecule, the result is excellent with vision improvement in the visual acuity as well as decrease in the macular. macular thickness. And in case of AMD, the average age which I found in my cases was around 72 years. And the visual acuity ranged from 360 to 6/12. The baseline surface thickness averaged to around 370 microns. That is around from 187 to 742. The vision improved in most cases and was stable in rest. The average I had to give was around 3.2 injection in 6 months. Roughly, I've done the study for around 9 months. Average gain in vision was 2 lines. And here is the case, 65-year-old female with best corrected visual acuity of 6.6/36, and post 3 injections, her vision has improved, and we can see the contour also has improved. The CMT has decreased, the intraretinal thickness has also improved. And this is the case of a BRVO, where again, post biosimilar, the vision has improved drastically from 6/24 to 6/9. We usually see such drastic improvements in RVOs only. And this is also a non-ischemic CRVO, where CMT decreased to almost normal in this case, and vision improved to 6/9. So in my cases, I saw none of the patient had any serious adverse reactions. So as we see in this, because our main concern, we might be tempted towards the cost of the drug, but our main concern will always be the safety. And if we see in this drug, we see the incidence rates has decreased progressively. In Feb 2017, it was 0.7. Then it was 0.1. And after, there was some recall of a drug also, which we have to be careful in using every biosimilar, because we have to see it over many years, how it responds, because it is something else to be there in a few case studies done in a laboratory and few case studies done, few incidents here and there. But then we need to have a collaborative study over years to know if actually the safety of the drug is there or not. So if we can see after that recall of drug, which they found out is due to the endotoxin, the incidence of the inflammation has drastically come down. And since Feb 2009, the incidence has decreased to 0.04%. This can be attributed to the higher pharmacovigilance by the company. And this is what we expect of the newer biosimilars, the newer companies which are coming into it, especially Reliance. As it comes into this field, we want a better biopharmacological vigilance of these drugs, so that we are more, we feel more safe with drugs. And my experience has been Resumab is economical, efficacious, and most importantly, safe. And it works as, in my experience, it works as good as the innovator. And it works across the indications. I've used it across all indications. I've used it in ROP also, because some babies come from, they don't get these drugs from they didn't get the drug, they didn't get the eccentrics. So I had to use Resumab in those cases, and it worked well. And it also avoids the medico-legal complications. And the main benefit is that it is sustained over multiple injections. And one thing I found interesting in this study was the ophthalmic biosimilars, the lessons from India, what we get is that there is a need of biosimilars, especially in developing countries, because the presence of compounding pharmacy is rare. It is an opportunity for the ophthalmic biosimilar manufacturers globally. And strict pharmacovigilance and the careful choice of immunological testing assay prior to market approval can counteract this. We have a perceived notion about this drug being inferior to the innovator drug with lower safety or efficacy. But if we have the stricter guidelines which validates these things, so we can be more confident in using the drugs, and especially talking about these drugs to our patients. Thank you so much.
Thanks, Urvashi. It was a huge number of cases. Yeah.
Urvashi, I want to know whether are you evaluating ellipsoid zone And external limiting membrane also continuity, discontinuity, and improvement with the passage of time after the drug. Anyone is improving. You are talking only of visual acuity and the central macular thickness.
Can you unshare while you answer the question? I can answer Sir's question.
Yes, sir. Sir, yes, I tried to see other parameters also, but looking at the whole the patient load we have, we honestly we don't go into so many details about every patient. But yes, I would like if I had a fellow like most people have these days. I would like to go into more details. Yes.
Dr. Pillai, you want to say something?
About the biomarkers in
yeah.
Okay. So the most important biomarker is visual acuity. That is point number one. Point number two, we have quantitative biomarkers in central subfield thickness or central macular thickness, etc. Number three is you are looking at qualitative biomarkers, and I would say that if you are looking at diabetic macular edema, if you have a submacular detachment, you are looking at some systemic diseases like hypertension, renal failure, etc. Regarding response to treatment, large single cystoid space do not respond as well as the spongy edemas and the other edemas. So you think that your number of injections will require to be higher. And if there is large cystoid macular edema or even submacular detachment, after treatment, the final foveal thickness will be a little less, which means that there will be foveal atrophy, which actually means that either there is ischemia or there is long-standing disease. So these are there. Then small hyper-reflective dots and all. People say that it is much more suggesting an inflammatory pathology, and people will move to Ozurdex. I have not actually strictly adhered to that. And regarding outer zone is concerned, obviously yes, the visual prognosis entirely depends upon the ISOIS junction and all those stuff, because that's the area of the cones. So if there's a problem there, obviously a visual acuity is going to be much lesser. And foveal hard exudate plaque, if it is there, again a very bad prognostic sign in diabetic macular edema. As far as AMD is concerned, I think similar biomarkers have been described. Multiple hyperreflective dots. Then there is something called, there are outer retinal tubulation. Like that, there are some biomarkers which have been there. But generally, the response in terms of central macular thickness overdrives all these qualitative biomarkers in AMD, because it is about fluid, presence of either sub-RPE fluid, intraretinal fluid, or subretinal fluid. If there is fluid, you inject. And maybe subretinal fluid, you can tolerate a bit. Pigment epithelial detachment, you can tolerate a bit. But intraretinal fluid, don't tolerate. This is the gist of what I think about biomarkers.
Okay.
So I think there's a lot to discuss in the question which Dr. Goel has asked, but we can go on to next presentation. Ruchir, can you?
Yes, sir. I'll just share my presentation. Is my presentation visible, sir?
Yeah, please.
Yeah. Good evening, everyone. Thank you, Ajay sir, Goel sir, Gupta sir, and NOS for giving me this opportunity. So I will be sharing a particular case with my use of Indian biosimilars. So there's no financial interest involved. So this patient is a 75-year-old gentleman who came to me in June of 2021 with complaints of mild blurring of vision in the right eye for the past 2 weeks. Now, he was both eye pseudophakic. His BCVA was 6.9 in the right eye and 6.6 in the left eye. I did an OCT and I realized that the patient had a type 1 CNV, and there's a low-lying membrane complex which is subretinal. which is sub-RPE. Then there's a lot of SHREM, there's a lot of SRF, and this is a classic CNV case with vision loss. So I advised the patient. By this time, I was purely using biosimilars. First, we were using Razumab, and since May 21, I've been using Vizumab. Now, Vizumab is, I think, same as Ranizurel. It is the same molecule being marketed by Cipla. So I've been using this molecule since then. So I gave the patient first injection of Vizumab. So this is Q2, BCVA improved to 6.6. There's mild SHREM still persisting. At Q4, BCVA is 6.6. There is no SRF now, no SHREM, and the disease seems to have gone away. At Q6, there is a very slight disease recurrence and very slight SHREM that is visible. And if looked closely, the height of the PD, the RPD also seems to be increasing a little bit. So this was a very early recurrence. At this time, as again, most of us here do not give monthly loading doses. Even I don't do that. So I gave the patient second injection of Vizumab. Unfortunately, patient presented 3 days later with a complaint of decrease in vision and floaters after the injection. His eye was clinically white. There was no conjunctival congestion. BCV had dropped to 660. There were 4+ AC cells, 1 millimeter hypopyon, lot of RL cells, mid-vitreous haze, and OCT could not be captured. Only thing that I could do was a fundus photograph, which is visible that there is significant vitreous haze. Although there does not seem to be any form of vasculitis, the disc is also looking healthy. So this patient was diagnosed to have a sterile endophthalmitis. I started the patient on oral Vizolone 60 milligram with a 10 milligram tapering per week. Luckily, the patient was non-diabetic. Topical difluprednate was started along with Homite, and oral Ciplox was given as cover. 1 week later, patient's BCV improved to 6/24, as is seen in the fundus photograph. Vitreous haze is much less. AC cells and hypopyon had also resolved. And this is the OCT picture. So if we look at the OCT, the haze is probably because of vitritis, but the CNV component of the disease seems to be quiet. There's no SRF, there's no IRF, and the molecule is working, but there was significant vitritis. So I notified Cipla about this incident. All current injection vials which I had were replaced with a new batch of vials. All other patients, I had to recall all other patients who were given the same batch injections. Luckily, none of them had any reaction at this time. So, sorry. So I had sorry. So this patient was all right till Q6. His BCVA was again 6.6. There was no vitritis now. And by 1 month, vitritis had subsided completely. Unfortunately, at Q10, he had a recurrence again. BCVA was 6.9. All signs of disease recurrence were noted. Now the question was, what was to be done? So what were the problems at hand? Should I repeat the same molecule or change to the innovator molecule? That was the first question. Second was, should I shift this patient to aflibercept? So regarding the first question, the patient was very hesitant. He had reaction with one injection. He did not believe in the same molecule, whether innovator or biosimilar. And he was also not very keen on the possibility of repeat injections every 6 weeks. Aflibercept conundrum is still there that for cashless insurance patients, Eylea for you program is of no help. So only if the patient is cash paying, patient will get any help from Eylea. So this happened in October, and by that time I had entirely shifted all my CNV patients to brolucizumab as a first-line drug. Now I started going through literature online as to what I could do in this particular case. And this was the publication that I found, where in a particular case of brolucizumab induced mild vitritis and inflammation, the authors used subtenon steroid and repeated brolucizumab with no reaction. So I discussed with the possibility of brolucizumab with the patient. I specifically told him about that there is a very high possibility that there could be a recurrence of the disease that he had. There was a possible benefit definitely in the robustness of the response of the other molecule, but the possibility of a reaction could not be discounted. So I planned to give the patient brolucizumab with 400 micrograms of dexamethasone. Now I did not give triamcinolone because the white crystals would have hampered visualization of the retina if something were to happen. A separate high-risk consent was obtained with the patient along with the standard brolucizumab consent, detailing the risk that the patient was about to face. And I prayed to God. So this is Q4 after giving brolucizumab 6/6, no intraocular inflammation, no disease activity. After this, the patient was lost to follow-up for 4 months. So he came back in April this year at Q20 with the BCV of 6/9 and the OCT shows mild disease recurrence. So the patient has again received brolucizumab and is due for follow-up next week. So my experience with biosimilars I've used biosimilars quite a bit. We were earlier using Razumab and now we're using Vizumab. There is definitely good clinical response, more or less similar to innovator molecules, as everyone has already discussed. So in the last year, I have used around 125 vials of Vizumab. I had 3 cases of IOI. One is presented here. 2 other had injections given along with cataract. So the company denied the role of injection in leading to vitritis. The company was definitely informed about all the cases, and I was surprised that at the end, Reliance Industries is the one who's making this molecule and supplies only marketing. So why this information did not reach? So had Dr. Prasad been here, I would have asked him. And in a lot of my diabetics and RVUs, I still give triamcinolone along with the molecule. So I think there is a possibility that a lot more vitritis could have happened had I not used steroids along with it. So in the same year, I used 10 vials of Accentrix, around 25 vials of Aflibercept, and 65 vials of brolucizumab with no intraocular inflammation. So although I've used this injection quite a bit, there have been cases, but none so far so severe that to deter me from using this particular molecule. Thank you.
Thanks, Ruchi. Very interesting. Because if I was in your place, I would be really hesitant to use brolucizumab. You've already had inflammation in the eye. Brolucizumab is known to cause inflammation. It requires a lot of guts. But well,
or you have to lose your hair a bit.
So, yeah. What do others say?
Richard, patient didn't agree for Accentrix also, because I would have preferred giving a innovative molecule in this particular case, considering that with most of the biosimilars we have risk of inflammation. You're very lucky, very interesting case, in fact.
Sir, actually, sir, again, the thing is that the molecule still remains the same. How do I convey it to the patient that with the same molecule of a different brand, reaction is not going to happen? There is no specific benefit per se of shift. The question that I was asking you, that whatever I've seen till now, there is no benefit per se of shifting to an innovator molecule and to assume that it will give a better response. Safety profile could be better, but there is no guarantee. And so patient could not be shifted to Aflibercept simply because he wanted cashless treatment. He was not interested in getting a reimbursement. He was not interested in anything. And there was a cap on his insurance policy that he would get only say 1.5 lakh rupee per year for injection. So had I given Aflibercept, all his policy would have been spent. Second thing was that, there is definitely, I mean, I'm not saying, I really got lucky, I think. I read this case report the same day I discussed it with the patient and nothing happened. And I mean, obviously things could have gone the other way also very easily.
So when you injected dexamethasone the first time, the next time when you injected Paginax, did you inject Dexa again?
No, sir. No, sir. Only the first time.
Invariably, with brolucizumab, the reaction may not come the first time, may come second time.
That's true, sir. But sir, I've used brolucizumab quite extensively now. I have patients who have received 8 injections, 6 weekly, 8 injections. There are patients who have received 6 injections. So personally, I have not had any brolucizumab-induced inflammation as of now. So I think that boosted my confidence. Plus the case report that I showed, it also somehow boosted my confidence. It might not have been the right thing to do per se, but because it worked, I'm actually presenting it. Otherwise, I wouldn't have presented it here.
Sometimes the circumstances make us take the decision rather than what we want.
True, sir. Aflibercept would have been my choice in this case, actually.
That's true.
Two submissions here. The first one is you had 125 injections of Vizumab in the last year, and you had 3 intraocular inflammations, which is fairly a high number. Okay, that is point number 1. And number 2 is we have post-injection patient coming with decrease of vision. That's a major concern that we should all think about and discuss about. Patient comes 3 days later with complete vitreous haze, hypopyon, you're not able to do OCT, disc is hazily visible, grade 4 haze. There are 2 possibilities. One is intraocular inflammation, and number 2 is infection. If it were to be an intraocular inflammation, if you give steroids, things would become normal. Or even if you don't give steroids over a period of time, generally things will become normal. But if it is infection, and if you don't treat it today, the patient will go into NOPL because you are injected directly into the vitreous. So how did you differentiate between endophthalmitis versus sterile reaction?
Sir, I would say there are 2 points that I would want to make. One is that the patient had a clean white eye. There was no pain, there was no lid edema, there was no clinical signs of endophthalmitis at all. Only thing was a mid-vitreous haze. There were no exudates that I could see. There was definitely hypopyon, but that's about it. So it was more like a post-cataract sterile reaction that we see in patients who have been tapered of steroid very soon. Only thing other was the vitritis component. Second is that when I started the patient, so I started him on topical steroids and I saw the patient next day. The patient started improving. There was no further decrease in vision and there was no change in vitritis. And the third point I think was that this was the time when we had seen a few similar cases of vitritis after cataract. So it did not look like infectious vitritis, infectious endophthalmitis to me at all. Had there been any other sign of infectious endophthalmitis, I would definitely have treated the patient with intravitreal antibiotics without fail. But clinically, it did not appear like that.
You gave topical or systemic antibiotics?
Sir, I did give the patient a topical antibiotic. I did give. I did not. I think I failed to mention it.
500 milligrams of ciprofloxacin, I think.
Yeah.
That's what you said.
Just because this was a very severe inflammation. So just to be on safer side, did you take an vitreous tap?
No, sir. Sir, I was fairly confident this was a sterile endophthalmitis. I had seen such cases before. And I mean,
okay, Ruchir, how many endophthalmitis have you had after injections?
Sir, I have seen a lot at RP center.
Okay.
And generally they happen within 2 to 3 days and they're in a pretty bad state.
Yeah, it's a very pretty bad state. Very rapidly it will progress. And usually they are white eyes to start with. And very rapidly vasculitis come in. And the whole media, within a day, it will become significantly bad. That is what I have seen. I have had 3 endophthalmitis.
And invariably, you will, if you have to save the eye, you have to do a vitrectomy.
True, sir.
So,
but
immediately.
If I find a patient like that, I'll probably inject immediately, tell the patient that I need to do a vitrectomy, and probably the next day or the day after, go ahead and do a vitrectomy.
Yeah.
Maybe
I think, sir, I will follow your advice only. I would have, seen this patient. Yes, we cannot actually make out in our mind whether this is infectious or non-infectious. Seeing the picture.
Sir, true, but the classical sees there are no vitreous exudate. So post injection endophthalmitis, what I have seen is actually very severe, and the patient would, even if it's a white eye, there would be exudates, there would be retrolental exudates, there would be a lot of other things also which did not happen in this particular patient.
The question is, you injected in the vitreous and you had a hypopyon. So that's something, you said post cataract surgery also you have hypopyon. Post cataract surgery you have hypopyon because you're operating in the anterior chamber. Injections of intravitreal you're giving in the vitreous cavity. So there, if you have hypopyon, you are lucky, or maybe you have to tell us the secret of...
No, sir, but let me tell you, sir, a lot of people now, post-injection, if there is an inflammation in biosimilar or Paganax, actually is treating the way Ruchir is treating, and everybody is getting away. And I think it is intraocular inflammation only. Only thing is, we are thinking that, you should think about the worst possible scenario and treat it. That is all. But I think this is, if out of 125 cases, 3 is a fairly high number, Dr. Prasad.
That is what I wanted to convey to Dr. Prasad, actually, that how has this information not reached him?
Because most probably the surgeons do not want to.
Which brand was it, Ruchir?
Sir, this was Vizumab. Vizumab, sir, the one being marketed by Cipla. And I expressly called them up. I spoke to their medical team. I gave them the patient details. I gave them everything. And I was told that this is being handled and everyone in the company knows. But somehow I think
I won't mention that the cases have not come to us, but were the cases of endophthalmitis?
Sir, this is sterile endophthalmitis. This is severe vitritis, severe IOI.
Okay. I suppose they have taken the batch number and everything from you.
I gave everything, sir. 3 cases I gave them. 2 they discarded, saying that because you have done cataract, this inflammation is because of cataract. This is the only case which was purely injection related sterile endophthalmitis.
Okay.
And Ruchir, you said
Ruchir, I hope our marketing team is also there in this Zoom. Okay, and the data has already come to us. Okay, because now I in this Zoom meeting I won't be able to identify the case, but I do promise that I will come back to you maybe tomorrow with the details of what have reached us, and then we will sort it out. Huh?
Sure, sir.
Dr. Ruchir, are you still using this drug?
I am out of stock now. There is no stock of Vizumab now. I finished my 125 vials.
Get Ranizumab now. Okay. So
either Razumab or Ranizurel now.
Innovator of Vizumab.
Get the innovator one.
True, sir.
So if you people can bear for 5 more minutes, should I make a small presentation?
Yeah, why not, sir?
Okay. It is actually not Can you see my?
Yes, sir.
All right. So this is one single case of DRBO and who we injected. So we have been using Ranitidine since last year, almost March, April. And this was a patient who came to me on 29th of July. He's a 54 years male, hypertensive, non-diabetic, and you can see a branch vein occlusion. Intraocular pressure, there is a slightly larger cup, but it was symmetric in both the eyes. It was actually sent to me, referred for injecting intravitreal injection of Ozurdex. I generally do not inject Ozurdex straight away in a patient who is phakic, and would definitely try anti-VEGF before a shift to Ozurdex. Patient was not on any anti-glaucoma medication. We discussed the options. It was slightly a VIP patient. I was keen to inject Eccentrix, but the patient had issues of economics and wanted to have a biosimilar. And I said, why not? And we discussed the options of the biosimilar. So we went ahead and injected a biosimilar. This was the picture of this patient at the time of injection. You can see in the in the superficial capillary plexus the area of ischemia. This is in the deep capillary plexus, and then when so this is how the picture looked like initially, and then when we injected, this is post-injection day 2, the visual acuity had improved to 612. You can see both in the superficial capillary plexus as well as in the DCP, there is some improvement. We went ahead and you can see here the velocity. There is a remarkable improvement in the macular thickness. And as Dr. Goel pointed out, there was, we definitely look at the EZ defects and we point out to the patients because that helps us in discussing the visual acuity prognosis on the long term. So this is on post-injection day 32. Patient was still doing well, and we normally in these cases we follow the PRN. Visual acuity was 6.9. At this point, he got a second injection of ranibizumab. He did well, and then he did not come to me for a really long time. His visual acuity was good because the outer retina was looking all right, but he had sort of fluid, cystic spaces, and this is 3 months post the second injection. So at this stage, he received a third injection of ranibizumab. And now, when we looked at this is 50 days post the third injection, the circularity of the FAC had improved. The ischemia and the SCP as well as DCP had reduced. So now you can see the comparative analysis of when he had edema. This is the octa between at the time of edema and subsequently. You can clearly see the change in, and in most of the cases of venous occlusion, we should ideally be looking at the ischemia at the level of the DCP. At this stage, we went ahead and did a micropulse laser for him. This is just to show that we can also have an alternative treatment with a 5% duty cycle in the macular area and a 10% duty cycle in the ischemia beyond it. Now the patient has been stable for 6 months. He has, so we have called him repeatedly. He is actually out of station post micropulse laser. So for learning for us was that biosimilars of ranibizumab was effective and safe in this patient. And we can combine these biosimilars with micropulse lasers in such cases to give our patients good results and reduce the number of injections. Thank you. Yes, any comments, or we can rapidly conclude? Gopal?
Sir, excellent, sir. All that ends well is good.
Yeah, all that ends well. So I think must thank both Dr. Gopal Pillai and Dr. Prasad for making this webinar very exciting. I think it was very informative. I think a lot of people are having wrong ideas that the biosimilars are not as good because they have not done as extensive clinical trials as the innovator molecules. Because invariably, this is a question which the medical reps who come with the biosimilars are asked that how many patients have you actually done? What are the extensive clinical trials? The take-home message is that for biosimilars, you do not need to do such an extensive trial. Even the FDA doesn't require it. It is, so they have to go through the stepwise process, which Dr. Prasad as well as Gopal clearly outlined. If that process is followed, we are basically concerned about the safety, efficacy, and purity of the molecule. And then comes the immunogenicity studies. And if we have all these in place, we don't need huge clinical trials. And that is where the cost saving is. Any comments by Professor Goel, Professor Gupta, sir, and anybody else?
Ajay, it was nicely discussed. The biosimilar, they have been extensively discussed, and we have learned a lot from these discussions.
Thank you, sir. Sir,
Sir, this is Dr. Prasad here.
Yes, sir.
So thank you so much, everyone, Dr. Arora and everyone, absolutely. It was really a discussion which has given even us a different kind of insight, a good insight. And as Dr. Gopal also mentioned, our brand, Reliance, is the innovator of Visumab. Yes, here, but the only thing is that I am sure that here the product Visumab is 100% identical to Ranizurel. Only apart from the storage and transport conditions. So for Dr. We will be in touch with him tomorrow. Ruchir, we will be in touch with him tomorrow, and we will take the details, as extra details as possible from him. And in terms of meeting all of you, I'm sure I make a promise that I will insist on the marketing team that I can meet all of you in your respective clinics in a short time. Thank you very much.
That would be great.
Thank you.
Any comments to help Gupta, sir? Yeah.
Ajay, I must thank you for this session. I've not spoken anything because I have no personal experience in this field. But it was a real treat listening to Dr. Prasad, Dr. Pillai, and other speakers. I have really enriched myself. And Dr. Pillai, this is first time I'm listening to you. And my congratulations to you. It's really great. I'm really thrilled about it. Thank you.
Thank you, sir.
Thank you very much, sir. I would just like to make a small point. Government of India is actually trying to make Indian biosimilar companies come on top, because all these companies, including Reliance and Intas and Lupin and even Sun is coming, Cipla, Zydus, all these companies are Indian companies who will offer biosimilars all across the world. And so government of India is basically trying to put it up by making a clinical trial network across India in which Indian biosimilars can be tested. And I'm leading that network. We currently has 6 sites. We are planning to increase it much further. So kudos to the Indian companies.
Yeah, Atmanirbhar Bharat.
Yeah, Atmanirbhar Bharat.
It's very good because, it happened in 2010, 2011 when Obamacare was introduced. It was with the introduction of the Obamacare that the guidelines for acceptance for biosimilar changed. So those guidelines have become the standard over a period of time. So thank you all so much, and I'm sorry for that we overshot the time, but I think it was worth it. Thank you all once again.
Okay. Thank you. Thank you, Ajay.
Thank you, everyone.
Thank you.
Everyone. Good night.
Good night. Thank you.
Good night. Good night.